Treatment strategies for the deficiency of adenosine deaminase 2. First, the definition. Deficiency of the adenosine deaminase 2 or ADA2, also called cat-eye syndrome, is the first molecularly described monogenic vasculitis syndrome that is an autosomal recessive disease. The ADA2 is caused by genetic hypomorphic mutations in the ADA2 gene that encodes the adenosine deaminase 2 protein. It should also be considered in patients with early set of fevers, rashes, and strokes, even if it is the absence of positive family history. Vasculitis and inflammations may affect internal organs, and the only way to diagnose it is by a genetic testing.
Adenosine deaminase deficiency, or ADA deficiency, is a rare genetic disease that results in severe combined immunodeficiency, or SCID for short. SCID can be caused by a number of causes, so this particular variation is called ADA SCID. Let's take a step back. Our cells have all the instructions of how to live and behave written on their own copy of DNA. DNA is made out of 4 nucleotides, which can do all kinds of cool stuff in their free time, like provide energy to various processes in the cell. Nucleotides are made out of a sugar, in this case deoxyribose, 1 to 3 phosphate groups, and a nucleobase, which can be adenine, thymine, cytosine, or guanine. So, the name of a deoxyribose-containing triphosphatic nucleotide, based on adenine, that makes up DNA, would be deoxyadenosine triphosphate, or DATP for short. These nucleotides are needed in equal proportions in order to make cellular division run smoothly. Now, nucleotides have a functional lifetime of their own, and our body has mechanisms on how to break them up into their building blocks to either be excreted or recycled. Let's focus on deoxyadenosine triphosphate. First, the enzyme adenosine deaminase removes an amine group from it, turning it into deoxyinosine monophosphate, or DIMP. Then, purine nucleoside phosphorylase comes in and removes the phosphate and the deoxyribose from DIMP, making hypoxanthine. Hypoxanthine is then oxidized twice by xanthine oxidase, first to become xanthine and then finally to uric acid. Uric acid can then be excreted by the kidneys in the form of urine. Now, one class of cells that divides quickly and therefore relies heavily on cell division to work smoothly are lymphocytes. Lymphocytes protect the body from pathogens like bacteria and viruses in 2 ways. First, B lymphocytes, or B cells, produce immune proteins called antibodies, which seek out and latch onto an invader, marking it for destruction by other cells. Second, cytotoxic T lymphocytes, or cytotoxic T cells, as well as lymphocytes called natural killer cells, go cell to cell looking for virally infected cells, or cells that look like they've started dividing uncontrollably, like a cancer cell. If they find a cell like that, they destroy it. So, simultaneous breakdown of both these pathways makes the immunodeficiency combined and severe, hence the name, severe combined immunodeficiency. Adenosine deaminase is encoded by a gene on chromosome 20, and typically mutations are inherited through an autosomal recessive pattern, meaning that the disease occurs when a child receives a mutant allele from both parents. Statistically, if 2 carrier parents were to have children, they'd have roughly 25% chance to have a completely healthy child with 2 healthy gene copies, a 50% chance to have a child that's also a carrier of 1 defective copy and 1 healthy copy, and a 25% chance to have an affected child with ADA SCID. In individuals with adenosine deaminase deficiency, a key enzyme, adenosine deaminase, is absent, and as a result, DATP cannot be degraded when no longer needed, so excessive amounts of DATP pile up. This huge excess of just one type of nucleotide slows the process of the cell division immensely. Imagine that you're assembling a pizza with 4 ingredients: dough, tomato sauce, cheese, and basil. It becomes hard when you have an excess of a single ingredient. For example, standing neck deep in tomato sauce makes it hard to find the basil, cheese, and dough, so that the pizza is made up of the right proportions. This is exactly the case here. Excess of a single ingredient, DATP, makes DNA replication extremely hard. And DNA replication is vital for cell division. This affects cells throughout the body, but particularly it affects lymphocytes because they are rapidly dividing cells. As a result, B cells, T cells, and natural killer cells numbers go down, collapsing the immune system. This makes affected children susceptible to any bacterial, viral, or fungal infections starting from birth. Prognosis in such infants is extremely poor without treatment. In terms of symptoms, because fighting infections takes a lot of energy, infants struggle to gain weight and are often diagnosed as having failure to thrive, and very sadly, many die. Treatment of adenosine deaminase deficiency usually centres on hematopoietic stem cell transplantation. The idea is to re-establish the immune system with donor immune cells that have the adenosine deaminase enzyme. Another option is to give enzyme replacement therapy through IV infusions of adenosine deaminase, but this requires weekly infusions and isn't curative. Alright, as a quick recap, adenosine deaminase deficiency is an autosomal recessive genetic disease resulting in a defect in genes coding for the adenosine deaminase enzyme. The result of ADA is increased accumulation of deoxyadenosine triphosphate, which interferes with normal DNA replication and collapses our lymphocyte immune system by disabling its proliferation. This leads to severe combined immunodeficiency, called ADA-SCID, which has a poor prognosis. Treatment options are hematopoietic stem cell transplants or lifelong enzyme replacement therapy.
Treatment strategy. Application in medicine. Steroids were mainly used as treatment, and after some time they had variable success. However, vasculitis and inflammation should be often treated with therapy. Drugs like azathioprine and cyclophosphamide have been used, yet with no success of curing it all. Vasculitis may cause headaches, fatigues, weight loss.
Well, we were interested in developing a new and improved version of gene therapy for a disorder called SCID. SCID stands for Severe Combined Immunodeficiency. It's a catastrophic disease in which patients have no immune system, the bubble boy syndrome as it's commonly known. This was an attempt to develop a new form of therapy that would be more effective than prior attempts, that would be safer, and that would be available to older children who have previously failed a standard bone marrow transplant. We saw very high levels of correction of the immune system at levels that are unprecedented in prior gene therapy trials. Previous trials have only corrected T cells, which are about 1/3 of the problem in SCID. In our trial, we found correction of not only T cells, but B cells, which make antibodies, and NK cells, which are another very important function of the immune system. It's the first time we've been able to correct all aspects of the disease. In prior gene therapy trials, often the patients still required lifelong antibody injections of what's called gamma globulin. In our case, we found that the subjects treated with our gene therapy no longer required gamma globulin therapy, which is both very expensive and people have to get this every 2 to 3 weeks. We'd like to extend this therapy to newborn infants, and we currently have a trial open at St. Jude Children's Research Hospital in which this would be offered as a curative frontline therapy. We believe that it's possible that this new form of gene therapy will offer them the best available current treatment for their disorder.
Application in pros and cons. Treatment with biologic TNF inhibitor was successfully controlling vasculopathy and preventing strokes in patients reported. Unfortunately, an standard care for stroke patients was recommended for them to end treatment with acetylsalicylic acid or anticoagulant.
In severe combined immunodeficiency, or SCID, the mutation destroys the enzyme adenosine deaminase, which results in damage to the T cells. There are 2 methods of gene therapy for SCID. In each, the role of vector is played by a virus that is harmless to humans. The DNA of a gene that codes for adenosine deaminase is placed in a plasmid and incorporated into a viral particle. non-functional T cells, or stem cells of T cells, are isolated from the patient's organism. These are then cultured with viral cells, which infect the T cells and incorporate a fully functional gene into them. Thus prepared, the T lymphocytes, or stem cells, are then reintroduced into the patient.
Pros and cons. Issue in Ecuador. In Ecuador, there are not a lot of epidemiological data about ADA2 deficiency, except for an article from students from the Universidad Católica Santiago Guayaquil, in which they determined that from 31 patients with extrapulmonary tuberculosis, they had an ADA lower than 35 units per liters, and also 5 patients with meningitis, and the 5 of them had an ADA elevated more than 6 units per liter. Also, more than 160 individuals have been diagnosed with adenosine deaminase 2 deficiency. Issue The conclusion of the research is to maintain the treatment of the TNF inhibitor, which, as said before, resulted successfully for decreasing vasculitis and inflammation, and for future therapies, use ADA2 protein or gene therapy in order to resolve it with specific quantities of therapy and finding a cure for it.